Today I'm joined by doctoral candidate Daisy Hoagland from the Icahn School of Medicine at Mount Sinai. As an early career researcher in biomedical sciences at this interesting point in history, Daisy's work has been overtaken by COVID. Of her dozen or so papers so far, almost all of them have been on COVID. Her work has added to the flood of crucial data on the pandemic, including the development of antiviral compounds. But how has this redirection of resources affected her educational and career goals? And how many other doctoral and postdoctoral students have been affected in similar ways? Let's find out. Welcome, Daisy.
Thank you so much for having me, Mary, and to the Eureka Sounds of Science podcast for having me. I'm excited to be here today.
We're really excited to have you. Thank you so much for coming. Can you tell me first about what drew you to a career in science, obviously before the pandemic?
Yeah, absolutely. It actually started in high school. I went to a vocational school in New Jersey called Biotechnology High School, and so it's like a public vocational school, and it kind of set me on the STEM track, and
Hmm.
I ended up going to the University of Vermont and studying microbiology. Um, and there, I realized quickly that I wanted to be involved in like actual lab science. So I started doing a work study position and eventually doing research for credit in Dr. Yvonne Jansen Heininger's lab, where we studied allergic asthma models in mice, and specifically reprogramming of metabolism in the context of asthma models. And
Huh.
so I was really interested in doing wet lab research, but after I graduated, I actually really needed to convince myself that I wanted to stay in science because I knew it takes like a lot to be able to spend like at least a decade of your life at the bench while you're
Absolutely.
getting all of these degrees. So I actually took a year and I did AmeriCorps Vista, um, which is basically I volunteered at a STEM mentorship nonprofit in the Bay Area of California.
Wow.
But
Okay.
basically, yeah. But then 2 weeks in, 2 weeks in, I learned a lot. Like I learned a lot of project management skills that I think are actually very useful for
Hmm.
like how I'm working now. Um, but 2 weeks in, I was like, oh my gosh, like somebody give me a pipette. Like I just felt uh totally out of my comfort zone. I needed to be at the bench. Or I
Hmm.
knew that I wanted to do wet lab science. So then I just immediately started applying to PhD programs that year. And then I got into Mount Sinai and I moved to the city. So
Wow. That's quite the crisscross of the country there.
yeah. So I definitely have a less specifically traditional path than a lot of doctoral students. I've always been on the STEM track, but I've also always been interested in a lot of other things as well. Um, but so because a lot of students they will do like go straight into a PhD program or they'll
Hmm.
spend a couple of years being a lab technician um, or a research assistant in a lab.
Hmm.
But so I just did it a little bit differently.
Yeah. I mean, I guess there's benefits to both of those trajectories, but in my opinion, it's always good to get some real world experience before you keep going with your education because it just makes it easier to figure out what exactly you want.
Yes, absolutely. And I think that having that experience like made me learn a lot of things about what like I do and don't want in a workplace and where I think that my work can be the most impactful.
Hmm. And of course, what you enjoy the most.
Right. Yes, also that. Yeah.
So, how has the pandemic affected your trajectory?
So mostly it's really just changed the specifics of my research, but overall I actually haven't been too affected, I think, except for just the acceleration
Hmm.
of my trajectory. When I so as a member of the Tanuver lab um at the Icahn School of Medicine, I was really studying like the antiviral innate immune response in
Hmm.
general, like just doing basic science research, and I was also doing a lot of uh, mouse work using for a viral engineering project.
Mm.
And so basically, when the pandemic happened, those were the two things that I was studying, and very specifically, it positioned me in a unique way to just transition all of the skills that I had learned doing, like in terms of understanding the antiviral immune response and being able to do small animal work. Um, I could use both of those to transition to understanding the host response to SARS-CoV-2 um, in a small animal model.
Mhm.
Um, and so it really was kind of chance that it just allowed me to take those things that I'd already been studying and use them there. And then definitely the past year has just been really accelerated. So just a lot of a few years
Yeah.
just kind of smushed into one. Um, but my trajectory has really stayed the same because I'm intending on continuing in academia, and after defending my thesis, starting a postdoctoral position.
So, what was it like as a doctoral candidate at your level when the pandemic first started?
It really felt like I was in a movie. Like, it felt like I was in specifically in the field of virology. I
Mhm.
was a second year in the middle of the second year of my PhD, so essentially that just means that, like, the entire trajectory of my PhD is still yet to be determined, because there's still several years left in theory. Um, and but I was acquainted with the lab that I worked in, like, I knew how to do assays, I knew how to run certain experiments. Um, so I had this whole opportunity of what the rest of my PhD could look like. And then when the pandemic happened and you are a virologist at a massive virology department at the epicenter of the pandemic
Mhm.
with a PI who is getting very involved in SARS-CoV-2 research immediately, and there's a BSL-3 just right down the hall from you. Um, it felt really uh, really surreal.
Yeah.
It still kind of does, honestly.
Yeah, I mean, I think it felt pretty surreal for just about everybody in the entire world, but
Yeah.
I can imagine being kind of right in the thick of that sort of base level science where everything is just kind of definitely taking off. I mean, we knew so little in the beginning that did it kind of feel like people were the whole world was like looking to you and your peers for some information?
Yeah, absolutely. It was, I think that that's part of what drove me to be able to get so much done, especially in the first year of everything happening,
Mhm.
that just the thought that something that I could do, not that any of us think that we're going to somehow by ourselves understand SARS-CoV-2
Sure.
or COVID-19, but like to be one tiny, tiny piece of the puzzle of contributing to our understanding of the virus or the disease, was definitely a motivating factor that got me through a lot of long days.
Yeah. So how long did it take for your lab to shift to mainly COVID work?
It was actually pretty immediate. So the first 2 weeks of March were like relatively normal. The second week of March is when everything started to like shut down.
Mhm.
Um, but I actually was one of the I got COVID like at that week.
Oh, no.
Um, yeah, so I basically I only knew this. I didn't have like severe COVID or anything
Uh-huh.
like that. It was a very mild case, but basically I had a cough for like a day. Um, and then I had a headache for several days after that. And
Mhm.
we realized that it probably had to be COVID. And then later on, I was tested seropositive for SARS-CoV-2. So basically, I self-isolated for um, for a week at the very beginning of the pandemic. But
Wow.
about but that week was when my lab entirely transitioned to SARS-CoV-2 work. So I came back and it was just like full force. Um, so essentially, like 2 weeks before the lockdown happened, a postdoc in our lab and a different graduate student started getting trained in the BSL-3 facility,
Mhm.
because no one in our lab had done BSL-3 work before, even though it was just down the hall. Because primarily, um, that facility at Mount Sinai was used for West Nile and hantavirus work.
Mhm.
Um, and so they were starting to get trained, but then by the time I got back, everyone was working just around the clock. analyzing samples, and everyone was doing everything in conjunction together for several weeks while we were really focusing on the first manuscript that came out of the Tenouver lab in April of last year. Um, and we were all just like working together to get it done. So it was very immediate. But the transition was a little bit starker for me because I left lab and then came back a week later, and the transition had just entirely happened.
Yeah. Oh gosh. So if anyone listening hasn't heard the term, BSL-3 labs are those that use or that are using or researching dangerous viruses. The only higher level is BSL-4. I'm not sure what BSL-4 specifically does, but I'm assuming things like Ebola.
Yes, that's exactly right.
Yeah, basically just
And,
probably just Ebola.
Yeah, Ebola. I think some Nipah viruses are BSL-4 as well. So some like
Yeah.
emerging viral pathogens, if you do them in animals, um, are also in BSL-4 as well.
Yeah. So can you tell me about some of these specific research you've done on COVID?
Yeah, absolutely. Um, so there are a lot of different collaborations that I've worked on, but mainly the work that I've done over the course of the past 15 months now, I guess, is defining hamsters,
Mhm.
the hamster model of SARS-CoV-2 infection. So for anybody who doesn't know, the reason that we have to use hamsters for SARS-CoV-2 is because mice don't naturally, the original circulating strain of SARS-CoV-2 does not naturally infect mice, which would of course be the go-to animal model
Mhm.
um, for in a lab to study any sort of disease. And so, of course, there are lots of ways to overcome the fact that they can't become infected with SARS-CoV-2 because you can introduce exogenous host receptors that
Mhm.
will allow the mice to be infected, but it gets a little bit tricky when you really want to study like things like tissue tropism and the host response, and you actually want to make sure that everything is exactly endogenously expressed.
Right.
Um, so the smallest animal that it became apparent does get infected with SARS-CoV-2 is the Syrian golden hamster. So essentially, these guys are about like uh, 5 times bigger than a bulb sea mouse when they're about 5 weeks old.
Mhm.
And you can put two in a cage, and essentially, no hamsters had ever been used at Mount Sinai before. So we just had to define uh, the hamster model to SARS-CoV-2 and just figure out how to work with hamsters in general.
Right.
Um,
Yeah.
yeah.
I'm just wondering, I mean, like you said, Sinai hadn't had hamsters previously. Do you happen to know how they figured out that hamsters could contract COVID? And I believe they even express symptoms similar to humans, like, for example, respiratory infections, things like that.
Yeah, absolutely. So there's a lot of early work done by Yoshi Kawioka's laboratory in Japan that did a lot of defining of the animal model. They had a paper that was published pretty early on, I think in June in PNAS, looking at hamsters. But besides that, we knew that because hamsters could be infected with SARS-CoV-1,
Ah,
like the original SARS-CoV strain. So people knew to turn to hamsters when this happened.
yeah. What were some of the other research tools that you've used besides hamsters?
Yeah, really, I think the most exciting thing about this past year, in scientifically for me, is that there's just this massive convergence of different research disciplines,
Hmm.
and it allowed the possibility for a lot of interdisciplinary work. So because everyone switched their focus, even people who didn't study microbiology or at all, um, wanted to pool resources together to see how we could study the virus using how they study science. So there was room for a lot of collaboration with people who use things like iPSC derived organoid models. So looking in vitro at cells that they're able to induce to actually resemble different parts of organs like alveolar organoids,
Hmm.
cardiomyocytes, neurons, intestinal organoids, with several different research groups. The work that I did surrounding that was with the Brennan's laboratory at the Icahn School of Medicine at Mount Sinai. So there's a lot of room for interdisciplinary collaboration as well. There was even room for collaboration with computational labs. Um, Professor Avi Mayan, who's also at the Icahn School of Medicine at Mount Sinai, did an in silico drug screen
Oh,
um,
wow.
using sequencing data. And then I was able to lead the collaboration from our side and test those drugs using that they were able to predict using their own computational prediction background. Um, as well as different sorts of collaborations with people that study things like anosmia, for example, with collaborators from the New York Genome Center. So, really, I think that the most interesting things in terms of tools is just the fact that I think we learned a lot about how tools from different disciplines can be used together
Hmm.
to really delineate some impactful things.
This is a little bit off topic, but I kind of feel like maybe the only, or one of the only silver linings to COVID was us all discovering how relatively easy it is with technology to collaborate with people who are at a distance from you. I mean, that kind of
Mm.
became the norm. Do you feel like
Mhm.
you like it sounds like you've discovered that collaboration has kind of ramped up during COVID?
Yeah, it definitely has for us. I can't speak for everyone because everybody has
Sure. Yeah.
different experience, and I know like I'm also talking about this as somebody who is a virologist.
Mhm.
Um, but it definitely has, and I think that has going back to the aspect of my trajectory and training has given me a lot of opportunities to see how different people from different fields think about different questions, and given me a chance to be able to like explain from a virologist perspective how I would plan this experiment or why we need to look at this, which I think has been a really valuable lesson and learning exercise as well that we can do in real life. And I think that also in terms of the science that SARS-CoV-2 research has brought about, I think a lot of the things we're learning about have a lot to do with probably many viruses. But
Mhm.
the fact that um, we can study these things on such a large scale, with so many people looking at them, is
Mhm.
just allowing us to uncover things about viruses and the immune response that really are wouldn't be possible to study without so many people working on them at the same time, and also having um, like human data,
Mhm.
um, in vivo data, and in vitro data to learn these things all at the same time with all of this funding um, and people working together to solve a problem.
Yeah, absolutely. So, but speaking of your trajectory, has this focus on COVID redirected your original career goals, or do you feel like you're still kind of on the same track?
Yeah, I'm still on the same track. I think that I went into my PhD not knowing necessarily whether I wanted to continue on in academia or
Mhm.
move to industry or move to something else as well. Um, but I always was open minded to everything. Um, I personally know that I'm going to stay on the academic track right now,
Mhm.
but I also know that for a lot of people that it has changed. Um, because of a lot of things. But I also think that specifically, I'm surrounded by all of these people who are in virology, and now, of course, there are a lot of new virology jobs that are opening up in
Mhm.
industry. Um,
Yeah.
so I think that can be appealing to a lot of people, and I know a lot of people whose intentional paths have changed um, because of the pandemic as well.
Yeah, that makes sense. Uh, could you expand, like, in your opinion, what has the pandemic meant for other early career researchers like yourself in this field? Like, what can you say from the ground in terms of what your friends are planning to do?
Yeah, so I think that it's meant a lot of different things to a lot of different people,
Mhm.
um, specifically depending on, their situation and their ability to adjust because of things that don't necessarily have to do with science, but just the things that they have to deal with in their life, like during the course of a pandemic, that allow them to like focus on science or not.
Right.
Like, for example, if you I can't imagine if I had children and this pandemic happened, and there are a lot of PhD students who do have children,
Mhm.
and trying to make this transition to do all of this research, I absolutely would not have been able to. Um, and I think as well, if you have family members who are frontline workers, and you're constantly or and also have at risk comorbidities for COVID-19 the past year, and you're
Mhm.
constantly worrying about your family, um, and of course those worries are even greater specifically in communities of color.
Yeah.
I think that for a lot of reasons, I was privileged enough to be able to take advantage of this opportunity in a way that a lot of other people in different situations wouldn't have been able to because of the toll that this pandemic has taken on how a lot of people um, live their lives.
Right.
Um, so I think that it's meant a lot of like heterogeneous things for different people. I think that the opportunity specifically in the field of virology, there's definitely a lot more now
Yeah.
because everyone's worried about the next pandemic,
Mhm.
and how the next pandemic is going to happen. So there's a lot of virology research, which is a really exciting time. Um, but I think that each individual has definitely had their own experience.
Yeah, that's definitely true. I mean, especially considering it's not as if COVID was the only thing happening in the world over the past year and a half, if only it had been.
Right.
Obviously, a lot of other things have been going on globally
Hmm.
since 2019,
Yes,
um,
that's true.
and before and forever. But if nothing else, and it brings more attention to virology and to, how easily these sort of pandemics can happen. I feel like that's good knowledge for us to have. And the more science that can be done around it, obviously, the better.
Yeah, absolutely.
Well, thank you so much for joining us, Daisy. We really appreciate hearing perspective from a researcher who's right in the thick of things.
Thank you so much for having me.